So, good evening, everyone. This is Dr. Satya Yadav, and it's a proud privilege to welcome you to the third webinar of Society of Pediatric Cell Therapy and Transplant. And today's topic would be on CAR T cells, which my chairperson and other people will give you the details about. But I'm as excited as you are to listen to the speakers, top-notch speakers who are here, and then a wonderful panel discussion which is planned thereafter about the scenario in India, how things are unfolding, and what's the potential. So it's all about cell therapy. That's we used to think is future will happen in India, but it's all happening now. It's very much present, and future is very bright. So I'll invite the chairperson for this session, Dr. Gaurav Kharya, and he's a consultant at Apollo Hospital, Indraprastha Apollo in Delhi. So Gaurav Kharya will take over. Over to you, Gaurav, for today's session. Thank you.
Right. Thank you, sir, for opening this session. So, as you highlighted, I mean, this is a very pertinent topic in today's day and age, and the way things are sort of growing in cell and gene therapy in India. And it's important both ways because few initiatives have already been taken by different groups. Dr. Gaurav Narula is here to represent one group. Sunil is here to represent the other, and there are some few other groups also. Apart from these beginnings, a lot still needs to be done, and to make it happen, I think we need to understand the basics of cellular therapy, basics of CAR T cell therapy. How do we sort of design the structure of CAR? Why is it relevant? Which thing helps in sort of achieving what particular objective? And that is that was the logic or rationale behind planning this session. And for this, we have none other than Dr. Tanveer, whom I'm sure a number of you might have heard in one of the previous sessions, or if not, then he will be speaking about the basics of CAR T cell designing and its clinical implication today. Although Dr. Tanveer has an illustrious research background, but just to brief about him, he's a highly accomplished scientist working at Jamia Millia Islamia, New Delhi, as an assistant professor. Over the past 5 decades, he has secured more than 10 patents in cutting-edge areas such as CAR T cell therapy, artificial intelligence and digital pathology, metabolic reprogramming, and CRISPR-Cas9 for genome engineering, with over 50 research publications in prestigious peer-reviewed journals like EMBO, Nature Communication, Scientific Data. His work is widely recognized. Dr. Tanveer completed his PhD at CSIR IGIB, New Delhi, and pursued postdoctoral research at NIH USA, where he specialized in receptor engineering, vector development, and CAR T cell technology. Since joining Jamia Millia Islamia in 2018, he has trained more than 30 scientists. A number of them are here with us today, who will be briefing us about their particular domain of work, advancing research in receptor biology, metabolic reprogramming, gene therapy, and CAR T cell, solidifying his position as a leading expert in cellular immunotherapy. Is an exceptional team of scientists working with him. Some of them will be interacting with us today during this meeting. So over to you, Dr. Tanveer. You can open your session. Thank you very much.
Thank you. Thank you so much, Gaurav. Can you see my screen?
Yeah, we can see you, but we can't see your screen as of now. So for the audience, please post your questions in the chat box, and we'll take all the questions, I mean, once the panel discussion is over, because what happens is that quite a number of these questions do overlap. So we'll note down all the questions and try to cover them together after the panel discussion, or along with the panel discussion.
Perfect. So, Gaurav, is it visible?
Yes, yes.
Perfect. So the topic that I have chosen for today's discussion and presentation is basics of CAR T cells. What are the designs which go into making the CAR T cells, and what are the current clinical data and understanding we have about CAR T cells? And with me, my team of scholars, researchers will be joining, who will be also presenting the work they have been doing in this domain. So basics of CAR T cells, I think now most of us who are here in this meeting have an understanding of what CAR T cells are. So T cells normally which are present in our body, they are not able to recognize the tumor cells. The reason being the MHCs which are involved in interaction with the tumor cells, they go down. The expression of these MHCs go down during the tumor conditions. One of the mechanisms is the molecules which are released by these tumor cells. They bring down the expression of MHCs. But then these cells, the T cells, although we have them in a large number in our body, but they fail to recognize the tumor cells. So then what we do is we engineer these T cells and we let them to express an exogenous molecule, which is a CAR on their surface. And these CAR molecules then are designed against a specific antigen, which is expressed by these cells, which we'll discuss in subsequent slides. And how the process works is we take blood from the patient in the clinic. The process is called as apheresis or leukapheresis. This blood is taken back to the lab where our team of researchers work on engineering these T cells by transducing lentiviruses which express these CAR molecules. And then that is what is called as chimeric antigen receptor T cells, CAR T cells. And these CAR T cells are first tested in vitro. That is the first validation we do whether these CARs are functional or not. Once we do this in vitro study, we see if the CAR T cells work against the tumor cells. Then we let these CAR T cells to expand to reach a certain number so that it can be an optimal dose for the patients. Once that optimal number is reached, these CAR T cells are then taken back to the clinic where the trained clinicians, bone marrow transplant surgeons, they infuse these CAR T cells back to the patient. And within the body of the patient, these CAR T cells then work against that particular antigen against which the CAR T cells were made. For example, the CAR T cells, CD19 CAR T cells, which are made against the CD19 antigen expressed by the B cells. So this is how the whole process works. And we have published a review article in 2022, where we have highlighted all the processes which goes into making a CAR T cell therapy, the simplest therapy which one can see, because it is the patient's own cells, and then these cells are made as the drug, which go back to the patient. So there are 6 steps involved in making the CAR T cells, as I was mentioning. Blood from the patient, engineered in the lab, then these T cells are taken back to the patient. This is how this simple therapy works. So what makes this approach a little complex is understanding of the molecular aspect of these CAR T cells, which is the chimeric antigen receptor, CAR, the molecule which is expressed on the T cells, so that these T cells are re-energized and then only they can recognize the particular antigen. So in the CAR, this is a basic design of a CAR molecule. The first component of this CAR is SCFE, which stands for single chain fragment variable. This is a domain which is derived from the antibody which was made against CD19. And it is only the SCFE part of this antibody, not the whole antibody, which was derived from that CD19 antibody clone. And then it was tethered with a domain which is called as hinge domain. So in addition to CD19, there are CAR molecules which are direct against other T cell specific antigens like CD20, CD22. And then for plasma cellular multiple myeloma, we have BCMA. For myeloid leukemia, we have 1, 2, 3, we have CLL1 and others. So what attaches these SCFEs to the membrane of the CAR T cell is there is a linker which provides the flexibility. And through this linker, these molecules are attached to the transmembrane domains. So linker is the hinge domain. Hinge domain have also been derived from the naturally existing molecules. Some of them are CD28 derived hinge, IG1 derived hinge, or IG4 derived hinge. CD8 is also used as a hinge domain. And then the other component of the CAR is transmembrane domain. So transmembranes are the molecules which make them stick to the T cell membrane. So most of the CAR molecules have transmembrane domains derived from either CD8 molecule, CD28, or ICOS. Then in addition to the transmembrane domain, we have, this is the orientation of all of these molecules which goes into the CAR design. We have a co-stimulatory domain. The co-stimulatory domain provides that persistence, proliferation, and effectiveness. And it is this domain which lets these CAR T cells to exhibit cytokine release, so these cells can persist longer, and release also activation. So these cells can then release GRAN enzymes and performance tract. So that is the function of these co-stimulatory domains. Most of these co-stimulatory domains are either CD4 28 based for 1BB, ICOS, or OX40. And then last domain which goes into the CAR molecule is signaling domain. Almost all the CAR molecules which we have now, 99%, they have CD3 zeta based signaling domains, because this is one of the best domain which initiates the intracellular signaling domain. Although we have tried and tested all the other combinations of co-stimulatory transmembrane hinge, but CD3 zeta has stayed as the same ubiquitous kind of domain, which provides that initiating signal for these CAR molecules to act. So this is now CAR design, and this is how, so this CAR design will define the generations of these CAR T cells. So back in 90s, we had the first generation CAR. That first generation CAR had SCFE, it had hinge domain, and then it had a CD3 zeta signaling domain. But that first generation CAR did not have any co-stimulatory domain. So what happened with the first generation CARs when they were tested in patients? They did not show any robust anti-tumor activity. And they also did not persist longer in the patients. Then came the second generation CARs. The difference between first and second was, in second generation, there was one addition, and which made these CAR T cells remarkable against all the B cell related malignancies. So second generation CARs have additional co-stimulatory domain, which is either CD28, that is a Yescarta product which is approved by FDA, or 41BB, that is the Kymriah product which was approved by FDA in 2017. So second generation CARs have one additional co-stimulatory domain, and then goes third, fourth, and fifth. Third generation CARs have two additional co-stimulatory domains, which gives them much better anti-tumor activity, and the cytokines are also better released in third generation designs than the second generation designs. So, and recent clinical data says that third generation CARs do outperform the second generation CARs because of the two additional co-stimulatory domains. And then fourth generation CARs are the ones which have a cytokine. In addition to the co-stimulatory, co-signaling domains, they have one additional domain, which is a cytokine. These cytokines, as we know, they are important in proliferation of these T cells. So once they go into the system, they should be always supplemented with a cytokine. So that cytokine has been designed to be provided by the CAR molecule itself. So one of the cytokine is IL-12. Now we have CARs which also secrete IL-2, IL-7, IL-15, and IL-18. So those fourth generation CARs are the CARs which also secrete cytokines. So these CARs will proliferate in the system, and they are also called as armored CARs. Then we have fifth generation CARs. So some group say fifth generation CARs are the ones which do have molecules which act as antagonist against the inhibitory signals, like the signals which are involved in the exhaustion of CAR T cells. I'll discuss about this exhaustion mechanism later in the slides, because one of the main challenges of lower persistence of CAR T cells is they exhaust quickly. So fifth generation CARs have tried to overcome those challenges by providing extra signals so that the exhaustion is taken care of. Then another group says that fifth generations are the CARs which have been modified by genetic manipulation like CRISPR-Cas or TALEN technology. And those CARs are also called as allogenic CARs. So Shakir will be talking about the allogenic CARs in the later part of this presentation. So where are we? Most of the data so far we have clinical data and understanding is based on the second generation CAR. Second generation and monospecific. That means this VH and VL, that is the SCFE domain, will recognize only one tumor antigen, which in this particular example is CD19. Since 2017, there are 6 approved FDA-approved CAR T products, which are all based on the second generation design. Now we can understand the design second, third. So all the 6 approved products are based on second generation CARs. Kymriah and Yescarta were approved in 2017, and then so on and so forth. So what is interesting is since last almost 14 years now, we have more than 34,000 patients who have received this CAR T cell therapy. And most of these CARs are second generation CARs. So having this huge clinical data, now what we have done is we have taken that clinical information, taken that data, and this is one of the examples of a meta-analysis where we have looked at all the clinical outcomes of these patients who have received these second generation CARs. They can be either 41BB, as in Kimria, or CD28 based co-stimulated domains, as in Yescarta. And then some of them are also third, fourth generation, but very few of those clinical data are available. So what we have learned from the second generation CAR T cell data is that they are remarkable for B cell malignancies. As you can see, overall survival is great. Event-free survival is also better. So more than like 40%, almost 40% patients do survive even after 5 years of receiving CAR T cell therapy. And even event-free survival is also impressive. But what it also tells us that 60% of the patients either relapse or they succumb to the disease, and they do not last longer when they receive any of these most of these second generation CAR T cell therapies. And they also, the data is also from the monospecific, where we have only one tumor-specific antigen targeted. And if we conclude the clinical data and what we have learned in these 14 years, we say that the two challenges. So I'm again talking here about only about the B cell malignancies. So the data suggests that in B cell malignancies, the relapse and if the tumor, patients who cannot survive for a longer period of time is due to two mechanisms. One is these cells do not persist in some of the patients. Second is, even in some patients, these cells persist. They are present in the system, but the antigen against which these CAR T cells were made, the antigen is lost, shed out by the cancer cells. And that is the challenge of tumor-associated antigen escape. So what that tells us that every patient is not like Bill Ludwig, who has also received a second generation CAR, patient to receive a second generation CAR, and who could live for more than 10 years. That means for more than 10 years, the CAR T cells were persisting in his body. Or Emily Whitehead, she is cancer-free since last 12 years when she received second generation CAR T, and the cells are supposedly surviving, persisting in her body. But every patient is not as lucky as they are. So that means 50% of the patients who will relapse, they have lack of persistence. So the CAR T cells do not sustain there in the body because they get dysfunctional or they get exhausted. The other challenge, as I was mentioning, is antigen escape. So when we talk about monospecific second generation CAR T cells, they will, even if they persist, but the antigen is not there. So that is the second challenge. CRS and ICANS clinicians have been able to manage this to a much longer breadth by giving the supplement tocilizumab and other drug molecules. Insertional oncogenesis has recently come up in the news, where a couple of studies published showed in bigger, higher tier journals. They showed that CAR T cell, because these CARs are introduced into the cells through lentiviral mediated process. So that can lead to insertional oncogenesis. And then again, limitation of CAR T cells is with solid tumors. But now we have some good preliminary data that CAR T cells do also work against some of the solid tumors, one of which will be presented by Ashi today against neuroblastoma. So insertional oncogenesis, there are reports. One is the latest publication in NEJM, although the cohort of the patient is smaller, 724. But in this study, what they have found is that there was one patient among 724 who had developed secondary malignancy. But that it was found later on after performing all the molecular analysis, that the insertional oncogenesis was not the cause, but it was due to secondary infections like Epstein-Barr virus-mediated T cell lymphoma. So we need to look at more data to see whether insertional oncogenesis any issue with the CAR T cells. But as we can see, the data, even if it is coming, it is even less than 1%. the benefit of this treatment far exceeds than the limitations which we can see. So talking about the other challenges which are persistent, like lack of persistence and antigen. So lack of persistence is a bigger challenge even in B cell related malignancies, particularly with second generation designs and also with the solid tumors. So our lab has been working on this particular issue since last many years, since my training at NIH, where I was involved in one of the products, Yaskata, which we made there at NIH. Since then, we have been working on this issue of lack of persistence. And we have tried and tested multiple methods since then. And then when I joined Jamia, my lab members were interested to take this challenge. And one of my PhD student, Arij Akhtar, she has come up with an innovative idea of doing metabolic reprogramming in these cells. And it has been her PhD work for more than 3 years, where she has used these metabolically engineered CARs to overcome the limitation of CAR-T persistence. So Arij will present some of the latest findings she has. Arij, are you there?
Yes.
Okay, over to you, Arij.
Thank you, sir. So, good evening, everybody. So today I will be presenting our work in the field of CAR T-cell therapy, especially focusing on tumor rechallenge and its effect on T-cell dysfunction and reduced persistence. So let's dive in. Firstly, to study tumor relapse, we developed an in vitro tumor rechallenge model, where rechallenge was done every alternate day. And we studied these tumor rechallenged CAR T cells for their function, mitofunction, the exhaustion markers, the anti-tumor activity, and their proliferation. So we found out that anti-tumor activity, CAR-T function, mitofunction, and proliferation was greatly reduced in tumor rechallenged CAR-T cells as compared to the control CAR-T cells. And upregulation of the exhaustion markers was also noted. Existing patient data was then analyzed and a downregulation of important genes like BECN1 and ATG14 was seen. These genes are involved in the autophagy and mitophagy pathways, and this downregulation of the genes was observed particularly in the memory T cells. So we hypothesize that this could be the reason of decline persistence. So next slide. What autophagy is that the cells have this special mechanism by which they break down dysfunctional organelles and get rid of them. So proteins like ATGs, they help to form a complex that aids in the autophagosomal engulfment and the ultimate degradation by the lysosomes. So for example, mitochondria. If it's a dysfunctional mitochondria, if it's a dysfunctional mitochondria, it needs to be degraded. So if autophagy pathway is somehow not working, it's dysfunctional. So these mitochondria will not degrade and they will accumulate over time and cause some dysfunction in these plasma cells as well. So next we studied the tumor rechallenge in vivo, and we got similar results. We found a decrease in the memory cells in tumor rechallenged models. And moreover, these CAR T cells also possessed an increased subset of T effector cells, but the exhausted cells, the T exhausted cells were higher in these T effector cells of the tumor rechallenged level. Next, what we did was we screened, we went through literature and we screened next slide, sorry. So next we dig through literature and we found 27 important compounds, and we use different combinations of these compounds to ultimately study the autophagy and mitophagy pathways. And we found a very potent combination of semaglutide, which is the GLP-1 receptor agonist, and urolithin-1, which increased autophagy and mitophagy of these tumor rechallenged CAR T cells. Next, we developed novel CARs that integrate the GLP-1 peptide, which is a natural receptor agonist of the GLP-1 receptors. Because semaglutide, it has unnatural amino acids, so it cannot be integrated into the genome. Hence, we use GLP-1 peptide. And we optimized its placement within the CAR sequence. And we selected the one particular sequence which secreted a good amount of GLP-1, had a good CAR expression, and triggered a good amount of release of CAM, which is particularly involved and important for autophagy. So we have also validated our results. Next slide. So all of these, the two videos here were just a brief insight into how all of this works. So we also validated these results in vivo, the integrated GLP-1 CARs. And we also found similar results with increased persistence, great anti-tumor activity, and good survival data, as you can see here. Next. So this is the gist of the study that we did, where we are comparing conventional secretory GLP-1 CAR T cells with urolithin A, and enhanced mitophagy and autophagy was observed in these T cells. Mitochondrial clearance was more, memory CAR T cell generation was higher, and these cells persisted for a longer duration of time, and the bonus on top is the anti-tumor activity. Thank you. Over to you, sir.
Thank you so much, Arij. And this work which Arish has done is currently being peer-reviewed on one of the journals, Cell Reports Medicine. And for this work,
and
we have
recognition.
filed
so,
patent, and Arish was
with
recently
that, our
awarded
first place winner
the
is
prize
Arish Akhtar.
for first
She's a
prize for
senior
this
research
work
fellow.
in
at
the conference which was held in Boston. So, so this is one of the mechanisms by which we have been able to undertake this challenge of CAR-T persistence by doing the metabolic reprogramming of these cells. And the combination of drugs which Arish was mentioning, we have tested a combination which led to 3,000 molecules we have screened, and among them we have found that semaglutide and urolithin A possess that activity to reduce the CAR-T cell dysfunction. The second major challenge which I was discussing is antigen escape. So this is a challenge when we have monospecific CAR T cells, CD19 CAR T cells as of now, direct against B cell, CD19 expressing B cells. So how to overcome this second challenge? For this, we have designed these bispecific combinations, which Dr. Rick will be talking about. He has made all these designs using a computation and AI-based method. Rick, are you there?
Yes, sir, I'm there, sir. So as Dr. Tanveer and Aris has already mentioned about the two major challenges of CAR T cell therapy. One is persistence, and the second one is antigen-associated escape. So to overcome antigen-associated escape, we have not only generated several different combinations, but also we have tried several different orientations of CAR T constructs, where we have used several different variable heavy and light chain, single chain variable fragments. With that, we have several combinations of the transmembrane domain as well as the co-stimulatory domain 1 and co-stimulatory domain 2 for the various constructs. So we have tried it in several different bispecific and trispecific CARs and generated 1215 CAR constructs. So which are computationally modeled as well as some of which are structurally tested. So computationally, we are trying to develop bispecific model that do not self-interact. Self-interaction is a major problem which leads to tonic signaling. So here we can see that two scFvs that are self-interacting and they are combined with a metal linker. So to overcome this type of problem of self-interactions, what we have tried to do is we have tried to mark down those key amino acid residues computationally and by checking their interactions with the receptor. And we have noted down those important residues and trying to mutate those residues to stop this self-activation problem of CAR T cells. As self-activation can lead to reduced persistence. To achieve this, we have used several different simulation models, and we have the final goal is to obtain is an optimum CAR design. And Dr. Nisha and her team, she have worked on AI ML based models where she have trained several different models based on the experimental data. And these models can determine from the sequence level, it can determine the tonic signaling score. And this tonic signaling score with higher tonic signaling score can lead to lower persistence in CAR Ts. So this can be in future used in designing several different bispecific and trispecific models, as well as this can be used in targeting the solid tumor. Over to Dr. Tanveer.
Thank you so much, Rick. And among the bispecific combinations that we have computationally designed, and then we have taken help from the AI, screened more than 1,000 of these CAR molecules in bispecific, monospecific, and trispecific combination. Finally, we came up with a molecule which is CD2019 bispecific combination. And this molecule was tested in vitro using the optimal this method of isolating 4 and 8 T cells. And once we have an equal number of these cells, we introduce this bispecific molecule through lentiviral mediator transduction into the T cells, and we checked Aries did all the work, where we looked at the expression of these molecules on the surface of the CAR cells. So compared to the conventional bispecific designs, our computational AI-based method led us to design a CAR molecule which has a homogeneous surface expression. So that will eventually take care of the aggregation of the bispecific and trispecific molecules which people have used so far in these designs. So this design was also tested in vitro using cells which mimic the same antigen escape mechanism, cells which are CD19 negative but CD20 positive. And also we have used a genome editing approach. This work was done by Dr. Shiva's lab at IGIB, where we have knocked out CD19 from these cells to mimic that antigen escape, and then CARs were tested against the CD20, which is expressed by those cells. We have tested in both monospecific, bispecific, and trispecific, and these CAR T cells showed much retain that anti-tumor activity, and then their persistence was also retained over time. This is one of the videos of lymphoma cells where we have tested these bispecific CAR combinations, and you can see over time how these bispecific CAR T cells eliminate the lymphoma models. So preclinical study for this work was done in collaboration with RCB, and first set of experiments were done 1.5 years back, and then the results were impressive when we compared this CD20-19 bispecific combination, and 2 co-stimulatory domains were used, ICOS and 4-1BB. And then it was repeated in multiple of those models where we show that CD19-20 CAR T cells exhibit much better anti-tumor activity, and then these animals could survive for a longer period of time. And interestingly, the cells, bispecific CAR T cells, were present in the circulation for a much longer period of time. So this is the molecule which we are working, and soon this molecule, along with Cellular Therapeutics, is being taken for the clinical trials. We are waiting for the final approval for clinical trials for this bispecific CAR T cell molecule. The other CAR Ts which we are working is BCMA CAR for multiple myeloma. And so the interesting part about this BCMA project is there are BCMA CAR T cells which have shown limited activity and tumor, this antigen escape has also been associated with the BCMA antibodies which are there. So, and these antibodies, existing antibodies to make the CAR for BCMA are not as potent as we have for CD19. So what we did is we thought to generate these antibodies, particularly CAR molecules for BCMA, using the existing knowledge of AI and computational methods. So in this direction, our lab has been rigorously working to find out new CAR designs using these AI ML-based models and computation. So this is the work which Varnit has done in the lab. Varnit, can you briefly tell us about what you have done for BCMA CAR?
Yeah, sure. So to optimize novel and optimize SCFEs for CAR T cells, we have used already pre-existing language models along with our in-house generated machine learning models. The process involves the sequence retrieval from the literature, where we get the sequence of antibodies which are pre-existing but not potent enough. So then we load them into our pipeline, which gives us a library of humanized SCFE sequences, along with the mutations that have the tendency to increase the binding affinity and stability of the SCFEs. The mutations were then incorporated to the humanized SCFEs and were validated structurally through molecular dynamic simulations. The best SCFEs from those were selected and proceeded to in vitro assays along with the mice model assays, which were done by Sheetal. And those show that the AI designed antibodies have better, if not better, then equally potence in killing the cancer cells. We are planning to take this work in clinical trials by the end of this year.
Thank you so much.
And over to you, Doctor.
Thank you. So this is the BCMA card which we have, the de novo AI-based computationally designed antibody, and the results, preclinical results are impressive so far. So now from B cell malignancies to the future for CAR T cells. So where are we now and what is the future? So CAR T cells for solid tumors has remained a challenge. And now we have a better molecular understanding why do these CAR T cells do not work for solid tumors. And although there are some reports, recent reports, which show that for some solid tumors, the CAR T cells do work efficiently. But the challenge remain for many more solid tumors. The designs that we are working on, I'm hopeful that in future we will have much better potent CAR T cells for solid tumors. One example is neuroblastoma, Ashi will briefly talk about. The other is manufacturing related issues, which is CAR T cells. When you make it, they are super expensive. Most of the patients in countries like us are not able to afford them. And the process which goes into this, making these CAR T cells, because these are patient-specific personalized medicines. So to overcome that, now we have good data across the globe, which says that you can make CAR T cells within the body of a patient. That is the in vivo CAR Ts. Within the body of the patient, these CARs can be made. So that will take care of the two pressing challenges. One is the cost of the CARs, because now these CAR molecules will be made within the patient. You do not need to have those super expensive instruments. Second is these CAR molecules will be available off the shelf. So you do not need to take blood from the patient and wait till the CAR T cells are made. These will be available all the time as off-the-shelf products. And I'm hopeful that by this year or by early next year, these in vivo CAR T cells will be tested in humans. So this work is being carried out in my lab, and Rukaya will briefly talk about the in vivo CAR T cells. First, CAR T cells for neuroblastoma. Ashi, back to you.
Thank you, Dr. Tanveer. As Dr. Tanveer rightly pointed out, there are certain limitations of CAR T cell therapy in solid tumors, be it the antigen heterogeneity, immunosuppressive tumor microenvironment, on target of tumor toxicity, or T cell exhaustion. There are certain challenges due to which we are not able to work in solid tumors. So I have been working on generating GD2 CAR T cells for neuroblastoma for almost 2 years now, and also EGFR variant 3 targeted CAR T cells for glioblastoma. So this is a representation of my work. Panel A suggests a general mechanism of killing of a tumor cells using CAR T cells that Dr. Tanveer has already explained to us. Panel B represents the manufacturing process that he has already told to us in detail. Panel C represents a list of antigens that are currently under review for neuroblastoma. So I am mainly focusing on GD2, which is a dicyalogangliocyte, which is present in the cells of the neuroectodermal origin during the fetal development. As a person grows, the expression of this antigen reduces on the normal cells, but it is overexpressed in the case of a tumor. So can you please go to the last figure, sir? So panel D represents the CAR T designs that we currently have in our lab, and the co-stimulatory domains we are using are CD28 and 41BB. So next. This slide represents my preliminary data validation, where we have validated these CARs in HEC293T cells. Panel C represents the percentage CAR expression of these CARs on day 5 of the experiment. Panel D represents the generation of a stable cell line, SHSY5Y mCherry. Panel E represents a cytotoxicity assay that we have performed in our lab. And hopefully we'll be going to the preclinical trials soon. Over to you, Dr. Tanveer.
Thank you so much, Ashi. And then in vivo CAR T cells, which is one of the upcoming therapy. And this work is being carried out by Rukaya. Rukaya will talk about the work on this in this domain. Over to you, Rukaya. Yes.
Thank you, sir. Hello, everyone. So I've been working on engineering lentiviruses for in vivo gene delivery for the last 2 years. And as already mentioned by Dr. Tanveer, the currently employed manufacturing process for CAR T is a cumbersome 9-day protocol, which often leads to loss of effector function by the time it reaches the patient. So the obvious solution is to make these CAR Ts in vivo. So for this, we have engineered lentiviruses such that It specifically targets the T cell and confers stability of these particles upon administration in vivo, yielding a functional CAR T cell within 24 to 48 hours. So as a proof of concept, we've been able to generate titers that are comparable to the conventionally used lentiviruses. And we've been able to check the transduction efficiency in mammalian cell lines as well. So we are hopeful that by the end of this year, we'll have a product that is ready for clinical trials. And we'd be more than happy to collaborate with clinicians who see a potential in this approach.
Thank you so much, Rupiah. And then allogenic CAR Ts is also an option where you make off-the-shelf CAR T products. So there are certain challenges with allogenic CAR T cells. Again, one of the challenges persistence. These allogenic CAR T cells have been shown to do not long last in the patient, and in some cases there have been reports of toxicities with the allogenic CAR T cells. But now our team has been working in this domain as well, along with Dr. Shiva's lab at IGIB. And Shakir has been carrying out this work on allogenic CAR T cells using CRISPR-Cas technology. Shakir.
Yes, sir. Good evening, everyone. So I've been working on allogenic CAR T cells targeted against B-cell malignancies such as B-ALL and NHL. Now, autologous CAR T therapies worked well in clinical trials, but it has its own limitations. So this is where allogenic CAR T cells come into play. And but graft versus host disease is a major challenge in allogenic CAR T cells. So I have been working on the graft versus host disease aspect of it. So I have knocked out TRACT and beta-2M to render it allogenic, and PD-1 to increase its persistence using mRNAs and modified guide RNAs, which support the multiplex T-cell editing much better than RNPs. Now, for the insertion of CAR sequences, I've been using lentiviral vectors as of now, but we plan to use targeted knock-in technologies in the future. Now, as you can see, the multiplex editing efficiencies in T cells using mRNA is much more promising than RNPs. Now, we have achieved up to 80% CAR expression in T cells using lentiviruses, and we observed similar proliferation and viability of edited T cells using mRNA as we have seen in Cas RNP edited T cells. Now, after further characterization of the multiplex edited CAR T cells using mRNAs, I should be ready for preclinical studies in a few weeks. Over to Tanveer.
Thank you so much, Shakir. And besides CAR Ts, allogenic in vivo CAR Ts for solid tumors, our lab has also been actively working on CAR NK cell therapy. And Sufyan, who is a PhD student in the lab, has undertaken this project, and he has been working on this project since the last 3 years now. Over to you, Sufyan.
So, good evening, everybody. So, as we have seen, a lot of work is being done on CAR T cell therapies. So, there are certain challenges in CAR T cell therapy, like cytokine release syndrome, graft versus host disease, and also the manufacturing process is quite cumbersome for the autologous as well as the allogenic setting. So NK cells are other alternative cells in the immune cells, so which can be used for the making of these CAR NK cells and to redirect them against particular tumor antigen. So NK cells have certain plus points, like they could be isolated from various sources like PBMCs, and they have a specific NK cell lines as well, and then cord blood and iPSCs as well can be used to make a homogeneous population of NK cells. Then these NK cells can be engineered with a CAR molecule to make them CAR NK cells. So what unique our approach was to make these CAR NK cells with NK specific co-stimulatory domains and as well as the transmembrane domains. So these are different from the conventional CAR T molecules which our lab and others in the world are working. So these NK cells are different. So we have designed a library, in-house library of around 36 CAR constructs having different co-stimulatory molecules. These molecules are based on the activating receptors of the NK cell as NK cells function based on the MHC independent pathway, based on the balance of the activating and inhibitory receptors. So these NK cells, when modified with having the NK specific co-stimulatory domains, we hypothesize that these NK cells would perform better as compared to the conventional CAR molecule. So what we did, we prepared this library and then electroporated these constructs into NK cells. For preliminary experiment, we used NK92 cells, and then we found out that these CAR molecules were able to express up to around 80% on an average, And then they also performed when as a co-culture against Raji tumors. So since we use bispecific CARs against hematological malignancies, so we use Raji cell which are CD19, 20, and 22 positive. So we used our construct were having 2219 and 2019 molecules as SCFP. So we found out that certain CAR molecules which had like co-stimulated domains specific to NK activating receptors like NKG2D, and also the signaling molecules of DAP10 outperform others in the co-culture activity as well. And we could also, here is a brief video of the NK cell targeting a Raji cell, which is in red color. So apart from that, we also have an mRNA-based approach for the rapid CAR NK cells. So this could be made like rapidly just by electroporation. So we have done the preclinical. We are about to start with the preclinical data for this. The in vitro analysis has been done. So we would be ready to collaborate for the future works.
Thank you so much, Sufyan. Thank you. So this is about what we have been doing in the CAR T cell space. What we believe as a group that although it's a personalized medicine, unless and until we come up with the in vivo CAR T, so we need to have robust clinical data with monospecific, bispecific, and new generation CAR T cells. So based on the patients, like more than 34,000 patient data which is available, we are also working on to develop these AI trained models to understand the transcriptome, metabolome, and proteome profile. And eventually we believe that the CAR T cells which have been sustaining in the in Emily Whitehead and which have shown a response even for more than 10 years, by understanding the molecular aspects of these T cells, we will be able to make CAR T cells more durable and with less off-target activity. And so they can actually then be called as living drugs based on the molecular understanding which we have. So future lies in getting more data and understanding these omic-based molecular signatures of these cells. Then we can have a specific patient-specific CAR T cell in future. So with this, I'll conclude, and I would like to thank Dr. Gaurav, who is a collaborator on the clinical part of it, and Dr. Shiva Prakash, who is a senior scientist at IGIB, and mostly the people who are there in the lab, driven, working all the time, an amazing group of scientists which I am privileged to have, and they are working there all the time. And I'm sure that we will be soon in par with the other countries who are working in this cell and gene therapy space. And in subsequent years, I am hopeful that we will take the lead in cell and gene therapy in the country than what world has been doing in this space. Finally, I'll be happy to have questions, and my group who's here, they will be also happy to take questions. Thank you so much.
Thank you, Tanveer and the team for this fantastic presentation, taking us through the basics of CAR T cell and also highlighting the sort of future approaches what you people have undertaken. And I'm sure, as you rightly said, in very near future, we'll be at par with any other country in the world in cell and gene therapy space. Now, there are a number of questions which are there in the chat box, and I'm sure there are other questions as well which are there in minds of the panelists as well. So we'll be taking these questions after the panel discussion, because I'm sure a number of these questions will be covered in the panel discussion as well. So please, Aries and Rick, Varneth, and everyone else, Ashi, just hang around because people might have questions directed to you also. Sorry, we could not introduce you individually, but I think I'll take a brief profile of all of you and we'll circulate it in the group, because you people have done great work for past 3 to 4 years. So clinicians must be aware of the kind of work which you are doing so that we can collaborate better.
Thank you.
Right. So Sunil, can we move on to the panel discussion?
Yeah, we can go ahead.
Right. So Dr. Sunil,
Tanveer, if you could stop sharing screen, then I will share my slide deck as well.
So yes, is it done now?
Yeah, yeah. Good evening, everyone, and first of all, thank you, Dr. Tanveer and team, for a amazing overview of the CAR T therapy scenario. And I think what was most impressive is that you already highlight the challenges which are there, and already I think you're working on the resolution or solution to those challenges. That I could see this 4 or 5 projects underway which are trying to address some of those challenges which we kind of are at this point of time facing in the CAR T therapy field. So we quickly move on to the panel discussion, and this is going to be more of a real time or real life experiences from India, and also what are the challenges we face at this point of time in India, and what are the opportunities going forward. So I'm privileged to have Tanveer again for this session, and Tanveer already has been introduced, and we heard him, and the amount of work is going on at his lab. We have also Dr. Prasad Iyer. Dr. Prasad is a senior pediatric hematologist oncologist and being specialist from KK Hospital, Singapore, and he brings in a lot of clinical experience in the field and in the real time use of CAR T therapies in the clinic. And we have Dr. Gaurav Narula, who doesn't need introduction to this audience. Dr. Gaurav is from Tata Hospital, Mumbai, and the principal investigator for the first CAR T therapy trial, which was a joint collaboration between IIT Mumbai and Tata Hospital, Mumbai. So welcome everyone, and thank you for your time for today's session. So I don't know, Dr. Gaurav, can you hear us, Dr. Narula?
Yes, I can, and yeah. I've been hearing Tanveer, and it was fascinating, really. Thanks.
So, Dr. Gaurav, I'm going to start the other way around. So, I just want you to, kind of brief the audience because this questions comes in very, very frequently. What's the status of CAR T approvals in India? What has been approved already, and what's available for use commercially is a question which already comes in quite often. So, if you could just brief us about that. And then we'll go on to the rest of the discussion.
Sure. So there are right now 2 approved CAR T cell therapies in India, and one is by ImmunoAct, which was a CAR T which was developed as a joint collaboration between us and IIT Bombay, which you just referred to, and that is approved for both relapsed refractory B-ALL and relapsed refractory B-NHL. Beyond second line for B-ALL and second line and beyond for B-NHL in patients above 15 years of age, based on the 2 trials which were conducted on the same IND. One was the pediatric population, which was exclusively on B-ALL, and the second was a phase 1 trial, which was on exclusively on B-NHL. Subsequently, the phase 2 adult study opened with B-NHL and B-ALL, and that completed approval, and that is the one which has led to the approval. The pediatric trial is still accruing, still enrolling, so hopefully we will be expecting approval sometime next year. The other approved product is by Immudeal, which is approved for B-NHL. I did not read the public announcement, but I think it is in adults only. For B-NHL, and it is not approved for B-ALL as of now. So these are the 2 approved therapies available. Ongoing clinical trials, there is more, but I think that's not your question right now.
Yeah, no, that's correct, actually, Dr. Gaurav, because it comes very often. I think you must be getting many phone calls as well. So, if I could summarize what you said is that the ImmuneAct product is for more than 15 for ALL as well as NHL in adults, and the Immunil, which was kind of trialed at our center, is for only for adults with NHL. So at present, actually, in children less than 15 years of age, we do not have any approved product yet. And of course, Tata is enrolling more patients, and we're also enrolling some more patients. But at this point of time, we don't have an approved product at this point of time, which can be used for children less than 15 years of age. Why I put this question? Because it's a pediatric audience and pediatric kind of session today. So we just thought we'd just clarify it here before we move forward. So thank you for that. I think this is for you, Dr. Tanveer. I think you briefly touched this a little bit, but I just wanted you to kind of, this question again comes in everybody's mind, that there are two kind of platforms, whether it's an open or manual platform or a closed or fully automated platform. So if you could just kind of quickly briefly mention it, and also what would be the pros and cons of each platform.
Yes, thank you, Sunil. The pros and cons of each, so both automated as well as manual CAR T cells have been tested in the patients. Both the products work absolutely fine. So the results are impressive in both these processes. So the challenge lies in the cost. So you have to have a balance between the cost and the efficacy. So efficacy wise, both are same, but the cost wise, the fully automated systems, CAR T cell generated, are super expensive. We have now more than 8 fully automated systems at this point in time available, starting from Miltini Prodigy to Cocoon to other instruments from GenScript. The cost is the only issue with the fully automated systems, and the manufacturing challenge, like you can make one product at one point in time, but that can be overcome if you have multiple of those systems available. So for manual method, the cost can be lowered down, but you need to have trained manpower for that. So across globe, we see the shortage for trained manpower in CAR T cell therapy. People who understand the processes of these, making CAR Ts, making lentiviral particles, and then using these manual methods. So I think product-wise, both are good, but it is boiling down to the cost eventually. That's why in India also, we are trying to focus on the manual methods of making these CAR Ts. But in future, I'm sure that we will have more people who are trained. Our lab has trained many people now. And then the CAR Ts they are making in the lab are, super efficacious. So, and we have also done quality control checks in the CAR Ts which we make in a regular lab. And then those CAR T cells do pass all the QC tests. So that means manual method can be one exciting area to explore, which will bring cost down. But then more people needs to be trained in this, and I think we are lacking that training. Extensive training aspect in it. Yes.
Thank you. So, Dr. Iyer, you basically have got experience in both. I know there's a lot of work which goes in Singapore as well, in both the platforms, and your collaborations with people abroad. So you want to expand a little. I think Tanveer has mentioned about cost, which of course is a concern, and that's one of the important. Of course, there's also cost of establishing a lab. Your manual can be done in a GMP facility, and that itself has got a cost. There's personnel cost and all that, which we do not sometimes calculate. But having said that, yes, in Indian setting, cost is an important kind of factor to factor in. But your thoughts on needle to needle time, scaling up, and efficacy. I think you briefed a little bit, but then your thoughts on in these factors when you compare the two two platforms, and what are you currently using in Singapore?
Good evening, everyone. Can you hear me?
Yes, we can hear you.
Yeah, thank you very much for inviting me. So, Dr. Bhat, very important question this is. So we have a closed system Miltony platform. Our needle to needle time, we prefer to use fresh produce. So we don't cryopreserve our product. So our needle to needle time is about 13 days currently. I know that China is doing the fast cards. I think their needle to needle time is as low as 5 days, roughly around 7-day mark. But their processes are completely different to ours. So that is the problem. And the second question I can see is the scaling up. So I'm sure Tanveer and his team will also say that, the bottleneck for patient care will also include the number of manufacturing that they can do, especially in the lab settings, and how many personnel are there, how many trained personnel are there. So that is going to always be the cost. Always going to be a problem. Coming to the cost, everything in Singapore is expensive. The car may be free, but the admission is not. The harvesting is not, and the 14 days of minimum admission is not. So we roughly quote about 250 to 300,000 Sing dollars for our patients. Also, although the car manufacturing is completely free, and this is the academic car. We also have the approval for using Kymriah in our center. We are quoting about $500,000 Singapore dollars for that, where we do the leukapheresis and send the product up to New Jersey. They will manufacture and give it back. So, but that cost goes to Novartis. It doesn't come to us. The $500,000 cost. Efficacy-wise, as far as I know, I don't know whether there is any head-to-head comparison between cryopreserved products versus freshly infused product. Both of them should work. I think generally the frozen products have a viability that is tested just prior to infusion, the CAR-T viability. And as long as they are to what our requirements and demands are, they should be the same. But I don't think there is a head-to-head comparison as far as I know. Does anybody else know?
So I'll answer that, Prasad. My question was with respect to manual versus automated. But cryopreserved, we use in a trial all cryopreserved products. So and as you said, there was a viability check before the infusion, and it wasn't. It was pretty good, and we in the whole trial the cryopreserved samples were used, products were used, and efficacy was as expected. It was done for a reason because in India we have to always think of kind of more of a model where you can manufacture at few sites and then kind of be able to supply to the end users at various locations, which may not be always possible in fresh infuse. That's why we kind of did a trial, you keeping that in mind, and so that we can kind of also have the proof of principle that cryopreserved products can be transported, shipped, and also can be equally efficacious. So just before we go to the next question, Dr. Iyer, I just heard that Novartis is changing their manufacturing platform, something called as Recharge, and the manufacturing time is going to come short, and costs are going to be dramatically cut down. You have had any experience with the new manufacturing platform they have kind of introduced, or not yet?
Not yet. We haven't had that. I hope the cost comes down, because I think most people can't afford these products anymore, especially in the West, if they have to pay out of pocket.
Yeah. All right. So coming to Dr. Narula again. So Dr. Narula, again, this is a very practical question, and as we have a lot of colleagues from clinical side joined this meeting. Which are the patients, in relapsed disease setting? Refractory, we can understand they have, kind of gone through various lines of treatment, but someone is relapsed. Which are the patients you'll send for CAR-T therapy? And, what would be the kind of message to the audience?
Right. I think very good question. So right now, it's a trial setting, so it's pretty much end of the line. So there's anybody end of the line is eligible for our trial. That means that they cannot go for any standard therapy left at that point of time, which includes transplant. But considering it, if it's approved, which it should be soon, I would say in India, very often second line is also last line. And we know for a fact that even in the transplant era, outcomes of high risk relapses, which we standardly, classify in a pediatric relapse rate classification, we do classify them into low risk, intermediate risk, and high risk relapses. Low risk being the isolated late extra admittedly and right to high risk, which is like early biliary relapse or intermediate ones, and some with the high risk cytogenetics. So now the high risk relapses we all know with chemotherapy alone, the outcomes are very, very poor. And even in chemotherapy, if you can get them into remission and take them through a transplant, their outcomes remain just around 30 to 40%. We are already seeing that CAR-T alone, even without subsequent transplant, is exceeding those kind of responses. So I would say high-risk relapses become a clear candidate for CAR-T even at first relapse. This is because the other alternative which is there, which is proven to be good and approved in the US, is actually Blinetto. Now, in these kind of patients, they would rather treat with some, induction, maybe half an induction or a chemolite induction, take up for blinatumomab, put into remission, and then take up for transplant. And these patients are now having 50 to 70% long-term outcomes, which was unthinkable, in the pre-Blina era. But if you don't have Blina available, which it's not available in many of our centers, we have unlimited access to blinatumomab. We are using it. Even then, it is nowhere near, meeting our requirement. But there is no plans for blinatumomab to be introduced to the market in India at any cost that which might be affordable in the near or even foreseeable future. So we are seeing a life without blinatumomab going ahead. So I would say all high-risk relapses should go for CAR-T. And if they can go for a transplant, if they go into remission with CAR-T, well and good. If they can't, they can continue on observation. And we now having better methods to refine who might need further treatment, after they go into remission with CAR-T.
So we will discuss that later. I think you answered the question second one already. That was, in low burden disease, not low risk, low burden disease. So Blina versus CAR-T. I think you mentioned both are options. Blina must have a problem is cost and availability, and that kind of makes it limited. But in low burden disease, that may be an option. But in a high burden disease or a high risk disease, it may not be an option as well. All right, so we'll go to Dr. Iyer again now. So Dr. Iyer, we just need to kind of briefly, I think Dr. Tanveer showed a slide on this already. Actually, that was the long-term outcomes. So unfortunately, most of the data which was initially presented was initial responses at 1 month, 3 months. And what is the long-term outcome? He has shown one slide, and if you could just highlight this and what's been your experience as well, you've been using for a long time of CD19 CAR, and also outcome in other platforms. Other platforms I meant is other other CARs, whether it is a CD22 or it is a bispecific CAR. So what are the long-term outcomes we are kind of expecting here?
Yeah, I think Dr. Tanveer alluded to this. I think if I think it all comes down to what facilities you have got, what is the pre-infusion factors that you have for your patient, what are your infusional factors like, and what are your post-infusional factors like. So in terms of long-term outcomes, if you look at any of the data, a lot of data is missing within the data that has been published. And I allude to that because I recently had a correspondence with the NEJM author of the TISA-Gen Leclucil paper. And I was a little bit horrified to hear that they had actually published data on long-term persistence, long-term outcomes as successful patients, but they had not mentioned how many infusions they had done for each patient. So basically, the devil is in the detail. If you general wisdom would be that about 50% roughly for a standard platform CD19 CAR, patients would be okay long-term. But then the results are a little bit muddied because some of them have had been consolidated with a transplant, some of them haven't been. Some of them have had repeat infusions of CD19 CAR, which they haven't mentioned in their papers. So the short answer to that is, if you have CAR T persistence on day 28 and day 90, you have long-lasting B-cell aplasia or lack of B-cell recovery, then in the trial setting, you can hope to keep them, just monitor them, provided they have no risks for, say, for example, lineage switch. In those kind of settings, in a very clear trial setting, you can watch them carefully with NGS level MRD. That is another
We'll talk
important.
about that for for post-transfer, what to do post CAR T. But other platforms, you think the data is better, same?
You mean for the dual CAR Ts?
Dual CAR Ts, CD22 CARs.
Yeah, I mean, I think CD22 on its own, single CAR use will be used mostly in patients who are CD19 negative. Generally, the recommendation would be for them to head for a transplant once the disease is under control. For CD19 and CD22 dual CARs in patients who are both antigens positive, in our settings, we are seeing some long-term survivors in the trial settings without consolidation for a transplant, but I don't think we have enough numbers in our settings. The biggest data is coming out from China, where they did the co-infusion of the 19 and the 22 with really good, excellent outcomes. Just the CD19 and the CD22 co-infusion alone. So I think that will probably obviously get rid of the antigen escape problem.
I know. I put this question to Tanveer now because he presented this, and Tanveer, Dr. Prasad highlighted this a little bit. CD22 by itself, poor persistence. Dual CARs was kind of thought to be the answer to all the questions. Doesn't seem to be. Sequential CARs or co-infusions seem to be a little better. But your rationale, I think, I just wanted to understand your rationale to still go with a dual CAR and a third generation, and how it's kind of different than what is the data already there published. So, I just want to scratch your brain a little bit on this.
Right. Dr. Sunil, as Dr. Iyer mentioned, so CD22, the data we have in many of the patients has been given to the patients who have relapsed with CD19 cards, right? And then they were given CD22. And for CD bispecific, whether it is CD1920 or CD1922, and then people have also come up with trispecific cards. So bispecific cards have different formats. So as Dr. Prasad was saying, there is a equal, so you have two different populations of T cells. One is made CD19, second is made CD22, and then both these cells are given together. So the other part is where you have tandem CARs. In one single cell, both these receptors have to be. So most of the data which, clinical data which we have, except the China, recent China data which Dr. Prasad is saying, so they had a tandem bispecific CAR, where one bispecific molecule is expressed in a single T cell. And that is the reason we have come up with a computation and AI-based approach. So when you have a single tandem CAR, which has 19 and 20, they do interact with each other. So the clinical data has not been as good when we have CD19 because of the interaction. So these molecules self-aggregate, they lead to tonic signaling, and then these cells do not persist for a longer period of time. But then this approach of China is good, where you have two different cells expressing these two receptors. But the limitation with that is, again, it will be costly because you are now making two different molecules. And then these molecules sometimes also have some...
so how are you addressing this, because yours is a tandem CAR. So how are you addressing the concern of exhaustion, competitive binding? How do you address that? I think you mentioned a little bit on that. So how do you address that?
So that's what Sunil, when I was showing the data which Rick, Vernith, and others have done in the lab from last many years. So we have used this computation and AI-based tool. So now think about, we have made a combination of 1200 and almost 40 molecules of CARs. So there's a huge library of CARs we have generated. with monospecific, bispecific, as well as trispecific CARs. So in our experience, trispecific CARs are the worst. Even if you change, you humanize them, you make a human body domain of SCFEs, they aggregate like anything, and then they do not pulse longer. You will see a basic signaling happening all the time in these trispecific designs. And among all these 1200 molecules which we have screened so far, finally we came up with a molecule which works perfectly fine. It does not self-aggregate. In a tandem format, it works absolutely fine. And then the design is unique in itself. We have a CD20 domain which is humanized, but where the CDR optimizations has also been done. So I think
So you mean the designing is such that you would would be able to avoid a competition between the two. And
Absolutely.
of course, one strategy which is there with dual car as in two different cars, and use them either simultaneously or in a sequential manner is another strategy which I think you mentioned.
And in addition to that, the co-signaling domain is also important. So dual car, with what co-signaling and stimulatory domain you are talking about? Does it work with CD28? Does it work with 41BB or ICOS? Or the combination of these molecules? So because that signaling domain with the bispecific configuration is so critical. That is why when you screen this large library of 1200, 1300 molecules, you get the answer, right?
Yes.
So, Sunil, if I may just ask, because Tanveer has put a very pertinent thing which we don't usually touch upon. So when you talked about co-stimulated domains, Tanveer, there are a number of co-stimulated domains. We are more familiar with 41BB and 28, but we saw in your work that you have been working on others such as ICOS, OX40. So how do you decide on your, yours is a bias, third generation with two co-stimulated domains. So how do you decide on a combination, of these co-stimulated domains?
Exactly.
To address the problems which Sunil has highlighted.
Absolutely. No, that's what we are talking about. The design, when you are making, it is 100, more than 1200 designs. So we have tested bispecific with CD28, bispecific with 41BB, bispecific with both CD28 and 41BB. And eventually we come up with one design which showed that ICOS in combination with 41BB is better when you are using this in a combination of bispecific. It does not show the same kind of activity when it is used in a combination of monospecific.
So it's basically different permutation combinations with regards to different co-stimulated molecules and with different spatial arrangements of the expression of the surface.
Exactly, Dr. Sunil. And that is what it is. Extracellular domain is equally important as you have the intracellular domain. It has to be a collaboration between these two domains which will decide.
So this is just on a computational analysis, Tanveer, or it has been, you have seen this efficacy, you have seen the superiority in your preclinical work as well.
In vitro data has been generated, and that's where we are training this AI model with these constructs. So all the tonic signaling methods assays have been done. And now Dr. Nisha, she is working on developing the final AI model. Eventually, the model which we are planning, and it will be released soon. So you feed any sequence of CAR, and you will get the outcome. What will be the tonic signaling? What will be the self-aggregation activation? And this is where this team is working on together. Dr. Nisha, Varnit, Rick, and others who have joined in our lab.
That would be cool, actually.
Yeah, absolutely. Looking forward to have that model.
So Sunil and Tanveer, can I button and make a comment?
Yes, please.
Please, please.
Absolutely, yeah. So actually, of course, this is fascinating work which has been done by Tanveer. I've heard it before, and I've been seeing it grow over the last 3 years, and really amazed at the development in a short time. One unforeseen problem which is there with the dual or triple engaging CAR-Ts is, apart from the dual, two cells separately induced with each, that strategy is different. But whenever you go for the tandem or the cis strategy, one unforeseen problem which came up when the in vivo studies is that, especially from the patients who got it, is that it's you really don't know which antibody is engaging, and whether the other one is functional at all when you do it. This is a thing which comes up. So, like, if you're getting a CD19-20 dual strategy, you don't know which one acted because most of the cells express both. That is one. And secondly, one may be preferentially binding over the other. So, at that kind of discrepancy, because the cells are lines, so you don't know which one did it, And why it matters is because the one which is exerting the pressure and the one which is not determines which escape is more likely to occur. So these problems are now being seen now in the dual CAR strategy. So just a word of caution that everything that looks great on computational biology or in the pre-clinic sometimes, gives unexpected results when you actually start infusing it in the patient. So this is one, and this doesn't get picked up in animal studies either, where the models are much more cleaner than what we see in human biology or in disease biology. So these are issues which are coming up. So I think the jury is still out there. I personally was a big fan of and fascinated by the dual infusion strategy, which I think Prasad would be in a better position to throw light on, because that's what RNDO was talking about in Singapore. That they see which antigenic expression is better and infuse that one. And sometimes they might infuse both if they feel like, but it's based on the profile of the patient and not necessarily as a go-to strategy, regardless of what's being expressed.
Yes,
absolutely.
So yes, so point
Sunil,
taken.
can I answer the first question, which Dr.
Yeah.
Gaurav?
Yeah, quickly, Tanvi, so because we have to
yeah,
move.
quickly.
We have
So
not already
this,
finished.
so when we are testing these bispecific CARs, Dr. Gaurav, we are looking at the cells which are CD19 negative, and then we and they express CD20. For example, when we have bispecific CD19-20, so those cells do not express CD19; they only have CD20, and then we see whether this CAR is working or not. And that is what I showed the data. We have done CRISPR-Cas based genome editing in these cancer cells, and you have made those cells which are CD19 negative, and it does work. And in second case, we have made a cancer cell which is CD20 negative. It only expresses CD19, and both show equal killing efficiency. And the expression wise, both CD19 and 20, we have antibodies to check CD19-20 expression, and both are equally expressed on the surface of the cells. I think that first question of yours, it is not just computationally designed molecules. Now in vitro and then in vivo, mouse models have been tested, where we have CD19 negative mouse tumor model. And then there also these bispecific CARs work absolutely fine. So that is the first part of your question, that we have validated these computationally designed CAR T cells in the in vitro and as well as preclinical models.
Sure. So
Yes.
actually, that's exactly what I meant, Tanveer, that the model is a clean model. So you will see the activity. I did not mean that the antibody is not working. I meant when you take two antibodies which work, put them on tandem and put them into a patient, you don't know which one killed the cell, because the cell expresses both to varying degrees. You also don't know which one is engaging more than the other in vivo when they're both on the same cell. So that's the concern which has come up from the actual patients who infused it. But we can take this discussion to a certain time. But I would just say that maybe you would like to look at that before you get into the clinical stage. It will be good. Yeah.
Absolutely, very well, valid suggestion. Yes.
Great. So, Dr. Iyer, you actually touched this a little bit. $1 million question. Role of transplant post CAR T, and I'll then pick Dr. Narula on what are the challenges we have here. But then, what is your take on this, and what has been your strategy? And I'm talking about actually out of a clinical trial. I'm talking about real life scenario.
I think this is a very, very $1 million question, as you just said. I think for conventional use of CAR T products, I think you have to take a composite of pre-CAR T infusion disease characteristics and post-CAR T infusion monitoring to decide whether you're going to take the patients for transplant. So for example, if somebody has a bone marrow blast of say 5%, somebody has a CD19 expression which is dodgy or dim, or they've had, say, for example, blinatumomab or inotuzumab before. These are patients who are going to be high risk for disease high risk for disease emergence. So these are patients you will take for transplant. Somebody with KMT2A, for example, they're going to risk a lineage switch. So those people, after the CAR T infusion, you will take them for transplant. Similarly, for patients whose CAR T does not last long post-infusion, say the 28-day and the 90-day mark are very important, where you look at the CAR T, you look at the B-cell aplasia, and you say, okay, these post infusion characteristics are very important to determine whether they need transplant or not. So these are the sort of main markers where we look at saying, okay, these are patients who will benefit from a consolidation of transplant. And most importantly, I think the more and more data is coming about next generation sequencing, MRD monitoring for these patients, especially post-CAR T. Any emergence of a small bit of MRD, you're already going to head into trouble.
True.
That
I
is
think
very
you
clear.
summarized it well. I think maybe one more thing is what CAR has been used. We see data from CD28. CARs, they all require a consolidation, and that's not the case with, for example, 41BB. So that also probably does play a role. I think there is a little. I think this you mentioned earlier. There's a little kind of algorithm, and you I think highlighted all the pre-transplant factors, which are genetic as well as the disease burden, post-infusion, whether it is MRD monitoring, B-cell aplasia, as well as persistence of the CAR. So these will determine whether a consolidated transplant is required or not required post-transplantation. So, Dr. Narula, very easy to say, and discuss on these platforms, but what are the challenges you foresee? Because I think, we discuss it very often, but there are challenges of CAR T transplant post-CAR T in India. So, what are your thoughts on this? Just in our context, Indian context.
The biggest kind of problem is affordability. And as you know, patients who have already reached the relapsed refractory status, even those whom we so traditionally call affording patients, many of them are non-affording by the time they relapse refractory, even for a transplant. And now we are talking about taking this population through CAR T, which, even though much cheaper than what it is abroad, is still pretty much out of reach and almost as expensive or even more expensive than a transplant as of now. Hopefully, it'll come down in the future. But at the end of that, to say that, okay, you need another transplant, we are pre-selecting out a very small population of people who will be able to go through the entire process. So I think apart from the fortunate few who will be able to take CAR T at the relapse refractory stage, and then also undergo and need and be able to undergo a transplant after that, we mind you, many of these patients may be relapsing post-transplant before they come for CAR T, and that's another population. Will they go back for a transplant after that? So we're talking about transplant CAR T, transplants, 2 transplants, and a CAR T. This is pre-selecting out a lot of people in the Indian context. So it's not going to work as a strategy for all, or even for most. It's going to work as a strategy for very few. So for most, I think it would be prudent to take patients, like I was saying, alluding to earlier, what we traditionally call high-risk relapses, would be better if they are guided towards CAR T from the time they relapse itself. Just spend some therapy to bring the disease burden down. Once it is down, it may not be eliminated, but it can just be low burden disease, and that's the stage where the West might go for blinatumomab. But unless if you have blinatumomab, go for it. But if you don't, better to go for CAR T and then wait and watch, and then try to see whether this patient will require further therapy after CAR T or not. And apart from transplant, we can look at alternate strategies like maintenance therapy, which has already been proved very well by Sarah Kurashian's paper. In fact, that was my original idea. In 2017-18, I thought that this is what we'll do. CAR T deke oral metademic chemotherapy dene ka after about 6-7 months or so, after it's been cured. Yeah, so the world is already doing that, and I think we need it more than they do. And CAR T is so expensive.
It may be more relevant for us to experiment it, actually.
Absolutely,
because
we should do it.
We don't have kind of much options once. And one of the other problems is, which we kind of also encountered, is that once it's hard for us to pick them very early, because NGS MRD is still kind of not in our clinical practice. By the time the disease comes back, you again have a little bit of a high disease burden, and then how to put them in remission again before transplant is another challenge, because they already had CAR T before, and most of them may have lost CD19 expression. You can't use another infusion if you already have kept it that time. So
yes,
a lot of challenges there. I think maintenance may be one option actually to target in our patient population actually.
Absolutely. So
Can I just add
NGS-MRD,
a small thing?
glad you touched on that. So we are looking at NGS-MRD now in our pediatric trial cohort, and so it's a secondary endpoint. So we are not making treatment decisions on it, but we'll have that data soon to help us guide the way forward. Sorry, Gaurav Kharya.
No, this is Prasad here. Just to add to that, I
Sorry.
think the Saragorashian paper was very, very good. But I think we should take that with a little bit of salt for now because the numbers are very, very small in those cohorts. And those were patients who had lost their loss of B-cell aplasia by 6 months. So if you have persistent B-cell aplasia, maybe you don't even need that maintenance.
Now, again, Prasad, for you is in and outside a clinical trial, we know what to monitor. It's very complex, but outside a clinical trial, in the patients you are treating outside a trial, so what do you monitor? I mean, there are some things which are practical to do, some things are not practical to do. So what do you monitor post?
So again, I'm going to put my Indian hat on and say that cost is a very important thing. So typically, we will definitely do a day 28 bone marrow along with the lymphocyte subsets, look at B-cell numbers. We look at CAR-T by flow at day 28. We will also do a 3-month one, very similar. If everything is okay between day 28 and 3 months, then we will do one at 6 months. And if money is an issue, we will do at 9 and 12 months. If not, we will probably not. If money is not an issue, we'll do it in 9, 12 months. If not, we will do it 6 months and follow that by a 12-month assessment, where we look at CAR-T persistence, we look at B-cell numbers, we look at NGS MRD, we look at immunoglobulins, because we don't forget the immunoglobulins. A lot of these patients will end up on IVIG. So these are our numbers outside of a routine, full blood count, clinical examination, all those parts.
Right. Siva, Sunil,
yes.
I am sure there might be a number of questions.
Yeah,
but,
so I will just put one question which is very important to Dr. Narula, and we'll then go to the questions. So that's last question. Dr. Narula, challenges referral. I think we've experienced this, and your data set, our data set has been very, very clear. The pre-CAR-T, patient status is extremely important. So what is the message you want to give here? Because as you said, it may be soon available in a commercial space as well, or even for the clinical trials. What would be the challenge referral, and what is the right kind of referral which should be made?
Yeah, I think that's very important, Sunil. We have both faced that, So, moment patients, people have a patient who needs a CAR-T or could be a candidate, there is that, okay, you just need to go straight for it. What I would say is that we already have a healthy experience from transplant. Why the rush? When you plan a patient for transplant, you take them through reinduction, you try to get them to MRD negative, you do a donor search, you get the HLA done. It takes 3-4 months for you to get them ready for transplant at the earliest. It's similar for CAR-T. CAR-T is not curing anybody by itself. It is the pre-infusion management. It is the pre-collection management. Then in between the infusion collection and the infusion, that management, then the CAR-T, and then the post-infusion management, and then the handling of the late complications, which we are seeing now, because now we're getting these long-term survivors. We're seeing these complications come up. So it's a combination of these 4 which is curing the patient. And any post-CAR-T therapy, which may be directed at the disease. So actually 4 plus minus 5, all 5 factors are going to cure the patient. So each one has to be planned carefully. I think the management till the point you can get the patient down to CAR-T, which is what you're talking about, the referral period. This is the most crucial part. I think once a patient is clearly identified for CAR-T, you should not go for very aggressive therapy. You should not go for anthracyclines or high-dose chemotherapy, which is likely to cause mucositis or gram-negative sepsis. The aim is only to bring the disease burden down and enough lymphocytes in circulation to do a collection. And we now try to do that. If there's a high burden disease at the time of referral, we say you can use VCR, steroids. Maybe you can add bortezomib or etoposide drugs which they have not been exposed to earlier. Inotuzumab is a very good way of
bridging prior to therapy is also very important before.
Yeah. So all these strategies, but don't go for hyper CVAD or something like that, and have a 40% chance of losing your patient right there. when you're gonna give them a live food induction. We are not interested in MRD clearance. Yeah,
I know. I'll stop here. I think we covered a few aspects, but there are some questions in the chat box. One or two we have already kind of answered, but one is for we can ask it to Tanveer because he's from the lab background. The question is there a difference in activity or efficacy of CAR T cells from pediatric versus adult in clinical trials or in preclinical work as well?
So I think, Sunil, this is with respect to the allogenic CAR T cells. So when you are making allogenic CAR T cells from a pediatric or from the adult, so there of course will be differences because the kind of divisions which a pediatric T cell has seen will be much lesser than the adult. So of course, I'm sure there will. I am not aware of any neck-to-neck comparison with the allogenic, but
I don't think we'll have pediatric allogenic CARs ever because you need it to be adult to be consenting for that. It happens in the unrelated donor scenario. So I don't think that is a valid kind of comparison we can make because we won't be ever able to do allogenic CARs from pediatric patients because they will not be able to consent for it. So I don't think that. But yes, there may be younger and older adults, and
I agree.
that will make some difference. The second question is, on Satyendra has been asking this question about refractory B-cell malignancies. He's asked you whether they have the same expression of those same markers which Arish was talking about.
So Arish, are you there?
You're asking Satyendra?
Arish
is not there. Okay.
Arish. So I guess these are the markers which are expressed by the B-cell malignancies. So they must be the same markers like B-cell malignancies, whether it is a refractory B-cell or these are the cells which are naive, they will have kind of same set of markers expressed on their surface, unless and until they are exhausted, fully differentiated.
And your thoughts on gamma delta CAR Ts? There's two questions on that. And there's some data for pilocytic astrocytoma. So what about gamma delta CARs?
So this is an interesting area which people have started exploring. We have not directly worked on gamma delta cells, but the data which we have seen so far is they are good in terms of taking care of the CRS. But now we have other ways of taking care of CRS. The limitation with gamma delta is the number of cells which you start up with is lower. So both pros and cons. Challenge is CRS, but you have many other ways to deal with CRS. Efficacy wise, both of them are similar.
Doctor,
differentiated other T cells are a little better than the gamma T cells in terms
I think
of.
there is a last question which I think is: are there any biosimilars for Blina around the corner? So as of now, I think there are none, but I'm sure that he's just waiting for the patent to get over so that he can bring
by next year
some biosimilar. Yeah, for Blina, he's almost ready with that. So I think as soon as the patent for the existing Blina gets over, within a period of 6 months, we'll have a few products in the market.
Absolutely, God.
So you don't need the patent to expire. So there are people working on it now. Since I'm going for a lot of these industry conclaves, they become 3-4 major companies are working on it. But see, just like a CAR T, you don't need the patent to expire. As long as you can have a novel antibody targeting CD19, you can make your own Blina and call it whatever you want. The problem comes in the linker. The linker which connects the two antibodies which is the most fragile. When it binds, it breaks very easily. So creating that linker molecule is where the challenge has been for making a molecule.
Excellent. Yeah, Dr. Gaurav, that is the self-cleave. There are linkers which get cleaved in the cell, inside the cell. So
Correct.
We have self-cleavable linkers and linkers which are cleaved with the proteases present inside the cell as well as outside. So now we have a repertoire of more than 10,000 linkers available, and where we are also making PROTACs. Ashi is working on one of the projects where we are making linkers which are resistant to the proteases within the cell. And one strategy for glioblastoma is our PROTACs, which Ashi is working. So yes.
So you need to get in touch. You need to get in touch with a few Sun Pharma and Pfizer and Emcure and sell them your patent because that's the challenge they are facing.
Yeah, absolutely. Those GLP-1 we have patented recently, and then PROTAC is also being considered for the patent. Yeah, but yeah, I agree. These are the challenges for the linkers when you make bispecific antibodies.
Prasad, last question. You can take this. Is pediatric cancers show better responses than of course older people. So the question is that why the pediatric carts are not kind of given higher priority in compared to adults. So I think we already give it. It's just that it takes longer for approvals for children than adults.
Absolutely. I think you answered the question already. I think the problem is also market-driven forces. Unfortunately, what drives the market for adult malignancy is much higher than what drives it for pediatric malignancy because the numbers are low. Secondly, I think, as Sunil said, getting approvals for these kind of products we are second in line after the adults, predominantly for safety reasons. And that has historically
the only product which has got approved in children first.
Sorry, come again?
So CAR-T was the only product was approved in children first.
Yeah.
Original or the first approval came for children, young adults, right? So
yeah, correct. Yeah. So, but historically, it's always been the problem.
Other way around.
So, yeah. But I think the way to get around this is how the CD19 CAR-T is now used for autoimmune diseases, for example. So that's how they expand the business.
True. So thank you very much. I think we have tried to address what was posted there, and thank you everyone for a very interactive and very kind of practical session. I'll hand it over back to Gaurav and Satyendra for the concluding remarks.
Right. So thank you, Tanveer, and more importantly, the rest of the team for taking us through the phenomenal work which you have been doing. And I'm sure we as clinicians will be benefited from the work which you and your team has undertaken. So over to you, Satyendra, to conclude the session and make people aware about the next one.
Good evening, everyone. And thank you to Dr. Tanveer, to all my junior stars, Harij, Rick, Rukaya, Varnit. You guys have inspired, I'm sure, a lot of junior pediatric hemato-oncologists today that you can dream, and you can dream, not only dream, you can do it also in the lab. So a special thanks to Dr. Tanveer and all you youngsters. Thanks to Dr. Prasad, to Dr. Gaurav Narula, to Sunil, Dr. Spai, Dr. Gaurav Kharya, each and everyone. And a very special thanks to MCure for doing it so seamless. The next, it's a promise, we will keep on bringing the, what should I say, the scintillating scientific meetings. And the next one is the last Tuesday on 29th of October, I guess. And we'll again meet. And we have kind of designed that every 2-3 talks onto the transplant, then the cell therapy. And today we have just touched based onto the NK CAR, and soon we will expand onto it, guys. So once again, thank you everyone for attending it. And all the youngsters, all the young scientists, thanks a lot from the bottom of our heart. Thank you very much, guys. Keep doing the great work. Thanks, Tanveer. Thank you, everyone. Good night.
Good night. Good night.
Thank you. Thank you. Bye. Bye.
Thank you. Bye-bye. Good night.
Thank you so much.